Hi everyone, welcome to the Panhandle Research and Technology Tour Plus, hosted by the Panhandle Research and Extension Center in Scottsbluff, Nebraska. This event was recorded on December 3rd, 2020.
Thanks again for joining us today for Part Plus, Day 2. And I've got the pleasure to introduce Mitch Stevenson. He's a range ecologist here at the Panhandle Research and Extension Center. And he's going to talk about a topic that even an entomologist can find interesting. He's going to talk about using targeted grazing to control cheatgrass invaded rangeland. So stick around for afterwards. We'll have a Q&A with Mitch. And roll screen, please.
Hello, today I'm going to talk about using targeted cattle grazing on cheatgrass invaded rangelands. This is a joint research project between the USDA-ARS based in Cheyenne, Wyoming, and the University of Nebraska-Lincoln, the Panhandle Research and Extension Center. Cheatgrass is an invasive annual grass that is present on over 20% of the acres here in the Nebraska Panhandle. It becomes a challenge as it can oftentimes outcompete native perennial grasses for resources and completely invade a region. The image with the windmill shows this, that shows a perennial grassland in the foreground, and in the background with the deep purple hue is a rangeland area that's been invaded by cheatgrass. The picture in the lower right hand corner shows a mixture of both cheatgrass, the purplish plant, and our perennial native grasses that are intermixed. There are a number of management options for cheatgrass in the western Great Plains. However, they vary tremendously in how effective they are, and none of them have proven to be a large-scale method to control this invasive species on rangelands. There are a number of herbicides that can be used. However, most of these herbicides require reapplication, and sometimes costs can be prohibitive in how to use these herbicides. Fire has been shown to reduce annual brooms in the western Great Plains. However, logistically, sometimes it can be challenging as well. There are a number of biocontrols that have been reported to be successful. However, recent research has shown that most of these bacterial amendments, soil amendments, are not effective in controlling cheatgrass within native rangeland environments. So spring targeted cattle grazing has oftentimes been championed because it is effective at reducing cheatgrass seed production and biomass, but it's also a way that producers can gain a benefit from grazing on the cheatgrass. However, there is a risk of grazing on native perennials early in the growing season. So the study objectives were to determine diet composition of cattle early in the growing season on mixed grass rangelands invaded by cheatgrass. We wanted to identify cheatgrass plant phenology that correlates with consumption by cattle and determine how early season grazing influences cheatgrass seed production. Now this is a 5-year study, and so we have 4 years currently analyzed. The research was conducted at 2 study sites, one at the UNL Experimental Range north of Scottsbluff, Nebraska, and 2 at the High Plains Grassland Research Center near Cheyenne, Wyoming. At each of the sites, paddocks were developed about 10 acres where 50% of the vegetation cover was cheatgrass and about 50% of the vegetation cover was native perennial grasses. So each year we had 3 to 4 yearling heifers that we turned out onto these paddocks. They were stocked at moderate stocking rates, and we also included GPS collars to identify the time cattle spent grazing in cheatgrass and non-cheatgrass areas. We typically tried to start beginning in mid to late April when cheatgrass was about 2 inches tall, and then we grazed on the paddocks until cheatgrass reached full maturity. We collected a number of different measures, including cheatgrass and perennial grass plant height and phenology, cheatgrass and perennial grass plant material for nutritional quality, as well as biomass every 2 to 3 weeks that we separated by plant functional group. We also took fecal samples twice per week, and we sent those to a lab to evaluate the fecal DNA within the samples to determine the plant species that the cattle were eating. This technique of fecal DNA sequencing has been done in a number of different studies, including studies to determine diets of bison, feral horses, cattle, as well as a number of wildlife species. The way it works is that they're able to extract the DNA and compare it with a library of DNA sequences from grass plants within the area. The results of our study, looking at the grass nutritional quality, so we have cheatgrass is the red dashed line, needle and thread, perennial cool season grass, and western wheatgrass, another perennial cool season grass, in the green and the blue dotted lines. As we can see, starting in mid to late April, the nutritional quality of all the species declines as the growing season progresses, which was expected. However, there were some yearly differences in how the plants responded. And if we look at 2017, we see that needle and thread and cheatgrass were fairly similar in their quality. However, in 2018 and 2019, cheatgrass tended to be a lower quality than the needle and thread and western wheatgrass. With the fecal DNA sequencing, we were able to estimate the proportion of each of the species in the diet of the heifers. And so as we see, there was some variability between the years, but the cheatgrass at some portions of time was selected compared to the needle and thread and the western wheatgrass. With this data, we were able to potentially identify key windows when targeted grazing could be applied, where they would select to a higher degree on cheatgrass compared to our native perennials. So preliminary results that we've looked at for predicting the consumption of cheatgrass include looking at modeling the plant height and flowering date. And what this figure here shows is 2017 and 2018, which we found were fairly good at predicting when cheatgrass would be grazed. So the circle there highlights the times when cheatgrass was most selected. And so that's when it was a mean height, roughly around 200 to 300 millimeters. And it was about 10 days, 5 to 10 days before and after the flowering event. The top 5 species in the diet averaged across the year include cheatgrass, needle and thread, western wheatgrass, as well as a rumix and a chenopodium species that were both forbs. Now the rumix species is also commonly referred to as wavy dock or curly dock. Those are the species that are out there, and they were present in our pastures. However, we do not think that they were at a high enough level in the pastures that the cattle could have that proportion of their diet be these species. And so we're still trying to figure out what is going on here, but we think it has something to do with the digestibility of some of these forbs and that they're being overrepresented within the diets with this fecal DNA sequencing. However, relatively speaking, we feel that the grasses are representative of the relative amount of the grass that's in the diet composition of these heifer species. When we look at foraging behavior, these figures show 2017 and 2018. And so the yellow dots signify when the heifers are grazing, and the blue dots signify when they're resting. And if we look at the graph, the orange line is when they're in a non-cheatgrass area grazing, and the blue line is when they're in a cheatgrass area grazing. In 2017, we see that as time goes on within the paddock, that the time spent grazing in non-cheatgrass areas tended to increase, and the inverse is true for the cheatgrass areas. It tended to decrease. In 2018, we did not see quite as clear of a pattern, whereas it started out with more hours grazed in the cheatgrass area. It tended to level out throughout the season, where they grazed fairly similarly in both cheatgrass and non-cheatgrass areas. So a big question is, was it successful, or does early season grazing reduce seed production of cheatgrass? And so we've only collected data in 2018 and 2019 that I have here. We also have data in 2020, and we'll collect again in 2021. But this image here shows the difference in 2019 between the grazed and ungrazed parts of the paddock. And so if we look in the grazed parts, it's clear to see that it's definitely set the cheatgrass back maturity-wise, as there's not nearly as much that's reproductive mature on between the ungrazed and between the grazed and the ungrazed. And we did reduce the amount of seed biomass year-end as we collected it by about half in both years. And so we are reducing that cheatgrass seed production. However, there is still a lot of seed, viable seed within the grazed areas that reaches full maturity. And so we're wanting to look into the future as to finding ways that we can graze more intensively on the cheatgrass at these key windows so that we can reduce even more of that seed biomass. It is definitely a marathon game that we're looking at. It's not gonna totally reduce the amount of seed in 1 year grazing event. So our conclusions was that our fecal DNA was successful in identifying major species available in the studied pastures, and we were able to identify most of the taxonomic groups. We did notice that forbs accounted for more than expected in the diet. However, we feel that cheatgrass consumption, even though variable by year, that from our preliminary results, we feel that we can identify key times for targeted grazing based on cheatgrass plant phenology. And with this information, we're hoping to move that into more of a very focused targeted grazing event that we can utilize and give suggestions to people on when they can graze the cheatgrass that have the most impact on reducing the amount of seed production. Thank you.
Thank you, Mitch. Thanks for that presentation. And yeah, thanks again to everyone for joining on. And I think we've got a bit of a Q&A session to engage in now. I think Mitch is on.
Yes, Jeff, I'm here.
All right. Good deal. So Mitch, I'll give people some time to think up some questions to post in the chat box or the Q&A bubble. I guess one question I've got off the top of my head really is, from a cow's perspective, they're looking across the landscape and seeing food, and maybe crude protein is a way to measure what they see as food in that landscape, and perhaps. And I'm wondering if there's a relationship between, the crude protein that you might see in the landscape in your analysis and, some of that GPS tracking that you've done. So do those, regardless of species, do those correlate to one another?
Yeah, we think so. And we have a lot of data. It's just it's separated out across the years, and so we're still trying to kind of get all that in the model. Essentially, is where we're at right now. And so yeah, that'll go into the model as well. That cow makes a lot of decisions every day on both where and what to graze. And in a lot of our perennial grass and even the annual grass systems, there's a lot of choices that they have to make, and so they have their preferences, just like we like to eat at our certain places, or we don't like to eat at certain places, maybe because of past experiences we've had at certain places or restaurants and whatnot.
Not to name
and so
any.
yeah, no, I'm not naming any, but cattle are essentially the same way. And so if they've had positive experiences with either a certain area or a certain plant species, that's going to have a positive feedback on them that they're going to want more of that. And so as that quality declines, so we have really high quality when grasses are really short, and as that quality declines, they're going to start looking for other species that may have a higher quality of forage that's there. It's just more palatable. They get a feedback. There is a feedback mechanism that some people have researched that tells them that, hey, that's good quality. We're getting what we need. Eat more of that. And so there's a lot of behavioral aspects in this, and because cheatgrass is in some years especially a very prevalent forage plant species that's out there, we want to be able to really pinpoint that time when it's going to have the most value to be grazed, but also to limit it and allow for some of those perennial cool season grasses that we want to see out there to be able to become more prevalent across the landscape.
Right. Yeah, no, that's interesting. I definitely have my food preferences as well. Jumping back a little bit, Not seeing any questions here yet. Thinking about cheatgrass control on rangeland and regarding the biological control tactics that have been attempted on rangeland for cheatgrass control. Curious what the major obstacles toward their success has been. Has it been a lack of establishment, or maybe inability to reduce cheatgrass stands, or some combination of both?
Yeah, for you mean some of like the biological soil amendments?
Yeah.
Yeah, so really there was a lot of hype in the probably the past 5 or 10 years about some of these, and a lot of that came from some of the research that was done in a controlled laboratory experiment where they found that, and a lot of these soil amendments or bacterias are within the soil already in a lot of places. And so, they amplified them in a controlled environment. They put them on seed, and they found that it really limited germination or reduced root growth under these really highly controlled experiments. This is the case with a lot of our biological controls. When you put it out in nature, you don't have those controlled environments. And it's often found that a lot of times these biological agents need very specific types of either climates or conditions to really maximize their full potential. And so even though it was seen as effective in a lab, there's recent research in the journal Rangeland Ecology and Management. They did a whole volume on it. All the research that has looked at some of these soil amendments across the Western United States, and they came away with a fairly clear conclusion, is that they just don't work in a field setting. And so to take caution, if and some of these are commercially available, just take caution in using that and spending a lot of money on that when you could use other things that have proven more effective.
Yeah, as you know, I do some work in biocontrol, and establishment is a big part of it, and there's a lot of factors that can go into successful establishment of any biological control agent. But then the other factor of that is, once it's established, can it meet the goals that you have in this case for this landscape? Can it suppress that plant species that you don't want there to the degree that that you'd be satisfied with as a range manager? I'm going to keep crushing away on these questions here, Mitch.
All right.
I've got a couple more for you here. I think the sampling technique that you use, the fecal samplings in your study, was really novel way, I think, to find really a high resolution kind of picture, if you will, of the landscape and animal behavior over that large area, which I think is pretty novel. And I'm wondering if there were any plant species that you didn't see represented in that sample that maybe you expected to see in the sample. So was there anything out there that they weren't feeding on that was sort of interesting?
Yeah, so that's a good question, and the answer is no. And so that's what led us to believe that we feel like it did a pretty good job of identifying the species, because we did a pretty extensive plant survey within that 10-acre paddock to make sure that we weren't missing anything within that area. Now there's still some kinks that we worked out of this technique. I think it's not perfect, but there's not really a great way to analyze diet selection unless you're going to follow a cow around and that cow allows you to get close enough to see every time she takes a bite what is being consumed. And so there are a few other methods that can be used, but this is a relatively novel, and the technique is now just becoming commercially available, that labs are doing it, so that we were able to send that out. And so we think it identified all the species that we expected them to eat, and especially for the grasses, I think it did a pretty good job of identifying the relative amount in the diet. And I say that because most of our grasses were pretty close to the same in quality and had the same general decline in quality as the growing season went on. And it verified what we expected to be, that we did have one warm season grass species that was prevalent in the paddock, which was prairie sandreed. And right about the time that we saw prairie sandreeds start to grow in early June, we started to pick that up in the diet, which we hadn't before then. So it leads us to think that it does a fairly good job of identifying which species are out there and which ones the cattle are grazing on. But the forbs especially, it just and I don't know if that's just the amount that's not being passed through in some of these forbs that they're not digesting. But it just over amplifies that DNA that's being picked up in that fecal sample to where it makes it appear to be more than what it likely actually is.
Yeah, it's a really interesting technique, and as it's refined more over time, it might be a pretty useful tool for research and maybe managers to gain insight as to what's happening on their land with their cattle. So.
Yeah, I think when we look at it from a toxic plant perspective, I think it could definitely have some management implications if you're seeing a lot of animals that are having issues with toxic plants, being able to identify when they're eating those, and other applications as well.
The last question I've got for you, Mitch, is regarding targeted grazing again. The goal of targeted grazing, I guess, would be to reduce the production of cheatgrass in this situation on a given pasture. What would the next step be for a range manager? In other words, what should be done, if anything, to manage these targeted sites to encourage desirable species to establish? In other words, the next step. So should we be successful in targeted grazing? Is there anything that should be done on those sites that would then encourage that maybe native grasses or native plants or forbs to establish?
Yeah, so I think if really targeted grazing requires, I think, an intense management to it in a lot of cases if you're really wanting to focus the grazing at the right time. And so we're grazing for about 2 months in this study, and really we're doing that because we want to identify what time is the best time. We wanted to kind of identify that. Now that we can make some of those models and kind of hit that time, the goal would be to graze it really fast at a high intensity and high stock density. So we're talking a few days maybe, and then pulling off of that paddock and allowing those perennial grasses to recover, because we know that they're probably going to eat some of the perennial grasses too, but giving those perennial grasses time to recover is pretty critical to making sure that we're not harming the perennial grass that's out there more than what we're doing in the cheatgrass, because we're just trying to get that cheatgrass seed production reduced. Now there are some herbicide options that are out there that could also be used strategically in hitting some of these cheatgrass areas, especially areas that maybe have a lot of perennial grasses already established in those areas. So you're kind of looking at those areas that are not completely invaded, but maybe are just in the early stages, that you can hit some of those with herbicides as well to kind of keep it in check. This is one of those species that has such a high seed production that if it is in an area, it's going to be difficult by using grazing alone to totally reduce it. But we think that this is a viable tool that a lot of people are already using to kind of help manage it and to set it back. So you're getting value from it as a forage, but also setting it back and allowing perennial grasses to recover more and more. So yeah, I think there's some management strategies that we're trying to work out and trying to understand as to where the best places this would be to work.
What would that management strategy look like? Are you talking about the ultra high stocking density, like a mob grazing sort of small paddock, quick movement, or more of a larger area, high density grazing?
Yeah, so it would be more of a mob grazing type of scenario. high stock density. I don't know if it would be mob grazing. I often look at as you're moving sometimes multiple times a day. I don't think it would be that high of a stock density, but it would look a lot more like there would be. And what we kind of found is that that window is a little bit. It moves from year to year based on plant phenology, which can be different year to year as to when that reaches the point, how long it stays at pre-reproductive, post-reproductive. Sometimes we're seeing a couple flushes in the same year. And so, yeah, the idea would be that you kind of target where you can hit it at that, where it's most selected. So it would be something in neighborhood of a couple weeks there, probably, that you would have really intensive stalking on targeted areas. And we kind of realize that this may not be something you can utilize across your whole or your whole rangeland, but it's something that you may just have to focus on for a few years on a very targeted location that you're going to hit that area every year at this key moment to help manage the cheatgrass in those areas.
Well, it seems like managers would have to be tied into, degree day development of those plants in some way to predict when they need to get out and target for a given year.
Yeah, so what we're trying to do is focus mostly on when it reaches reproduction, and so it would be more of a visual type estimation where it's as you see it reach this point, that curve as it matures, that this is the time to get on, and then this would be the time to pull off of those pastures.
Mm-hmm. Really cool, really interesting work. Let's see. Got a quiet crowd this morning, and that's okay. So again, appreciate everyone joining us. Mitch, thanks for the Q&A and follow up. Looks like there aren't any other questions. Mitch's contact information, email, even a cell phone number. You can text him anytime.
Yeah.
And yeah, thanks for the presentation and the information, Mitch. And